some of the burning questions that you all deal with about aging, and particularly about the impact of the menopause on the female brain and its risk for Alzheimer's disease. I first wanted to just acknowledge that we have had a long-standing collaboration with colleagues here in the School of Gerontology, with Christian Pike, who will be speaking after me, and Todd Morgan and Tuck Finch for many years. And this has been a very productive collaboration. I won't be talking about this work today, but we will be having our own presentation on that soon. All right, so what about Alzheimer's disease? I think many of you know that Alzheimer's disease is increasing exponentially, and that it has a huge burden of cost, both in terms of health care and the care that is required for individuals with Alzheimer's disease. And this is the face of Alzheimer's disease. It's a female face. 68% of all individuals with Alzheimer's disease are female. And we think that the way that the female ages will provide insights into understanding the risks for developing Alzheimer's disease in women and in the men that will develop Alzheimer's disease. So our question has been for many years, what is unique about the way that the female ages that generates a vulnerability for developing Alzheimer's disease? So as I mentioned, 68% of all individuals with Alzheimer's disease are women, 32% are male. And one clearly initially asks, well, is that just because women live longer? Well, when you look at the average age of individuals in the United States, there's about a 5-year age differential between the life expectancy for men and women. Not so much as you would anticipate that would then translate into 2/3 of the individuals with Alzheimer's disease being female. And then when we look at, again, at the youngest ages of individuals developing Alzheimer's disease at age 65, this is regrettably not very clear, but at 65, where there's not a huge differential between men and women, those who have survived until the age of 65, what you see is that there's almost a double risk of developing Alzheimer's disease if you are female that continues out across subsequent decades. So we have really focused on what is it about the way that the female ages and that may, in fact, preclude her to a vulnerability of Alzheimer's disease. Okay. So we have focused on the bioenergetics of the brain, and I'll tell you more about that. And I'll give you a summary of our research focus and some of our recent and ongoing studies about how the brain both generates its energy and utilizes that energy, and our translational efforts to translate our basic science knowledge into therapeutics to prevent Alzheimer's disease. So a quick spin around estrogen in the brain, and this is true for the female brain as well as the male brain. Both estrogen and progesterone will promote the generation of new neurons in the brain and their survival. Estrogen in particular promotes their connectivity, and this is exemplified in the development of new morphological points of contact, or dendritic spines. This is also exemplified in an increase in synaptic transmission in the brain, and in this, what I'm depicting now is the area of the brain that you're using most extensively to process information, and which you will utilize to encode that information over a short period of time. And then you will transfer that information into the cerebral cortex for long-term memory, at which point we will then test you to see whether you actually made that transfer. That was a joke.
They just had lunch.
In addition, estrogen is regulating the energy producers in the cell, the mitochondria, and that's what I will be focusing a lot of my talk on. And in some, estrogen promotes the generation of new neurons, the generation of new neural circuits, and the ability of those new neurons to integrate into neural circuits to promote learning and memory formation both in the hippocampus as well as in the cerebral cortex. So that is about 30 years of basic science research from my laboratory and a host of other laboratories. And the other take-home message that we have gleaned from our research is that there's a healthy cell bias of estrogen action, that if you treat neurons with estrogen prior to degenerative insults, there's an increase in survival. If you instead treat them and create diseased neurons and then treat them with estrogen, there's no beneficial effect, and there may be an exacerbation of the degenerative process. So estrogen really does have a healthy cell bias of action, and that has led many to propose a window of opportunity for estrogen therapy and for the benefits of estrogen therapy. This is a quick spin around the mechanistic analyses that we have conducted over the past 20 years trying to understand estrogen action. And the take-home message for us was that estrogen mechanisms of action converged upon this organelle. We kept thinking it would converge upon the nucleus, but in fact it converged upon the mitochondria. And the mitochondria is, as you know, the energy-producing organelle of the cell. So let's talk about that energy a little bit. The human brain constitutes 2% of your body weight and consumes 20 to 25% of your energy. In fact, your brain right now, if you're awake and listening and encoding, your brain in 1 minute will use as much ATP as your one leg will require to run a marathon. That's a lot of energy. And you can already, I hope, see that the energy, the brain being an energetically very demanding organ, is also vulnerable to declines in energy. So, what is that energy being used for? It's essentially being used to reset the channels that determine the membrane potential. And what does that actually mean? That means that it's hitting the reset button that allows your neurons to function rapidly and repeatedly. So imagine, what would you propose is the first sign of a debilitating condition that targets a decline in ATP? Would it be running a marathon? No. It would actually be thinking, processing, encoding information. And that is indeed the first symptoms of Alzheimer's disease. So let's look at what those organelles that are producing ATP look like. And how many of you have seen mitochondria in a biochem text or a biology text? Yeah. Pretty boring, aren't they? Well, not really. So here are neurons that are labeled with a dye that labels mitochondria. And what you see here are the neuron cell bodies, here and here, that are loaded with mitochondria. And what I hope you can see are the neuronal processes that have these very dynamic organelles that are moving throughout the neuron to generate energy wherever it is needed. And that's a very low density of mitochondria. And here's a little approximation, a very weak approximation of the mitochondrial density in a neural circuit in your brain. So you can see, first of all, that there are thousands of mitochondria in your neural circuits, and that's replicated many times. That can't be true. 5 minutes? Okay. All right, we're going to speed right along like these mitochondria. So this is work that was done, that was the doctoral thesis work of Ron Irwin in my laboratory. What he basically did was a proteomic analysis of estrogen regulation of the mitochondrial proteome, the proteins involved in mitochondrial function. And the take-home message from that very, very important work was this: that estrogen is promoting the utilization, the metabolism of glucose. What's not shown here is that it's also increasing the uptake of glucose into the brain. Now, why is that important? Because glucose is the primary fuel of the brain. It is the primary fuel of the brain. Your muscles can use other fuels like fatty acids and ketone bodies, but your brain is not your muscle. And I'll tell you about why that is in a moment. And in addition, estrogen will promote the conversion of this glucose metabolism byproduct by pyruvate into the mother of all molecules, acetyl-CoA. And why is acetyl-CoA important? Acetyl-CoA enters into the Krebs cycle or the TCA cycle to generate electrons that now move that are involved in oxidative phosphorylation and ultimately the generation of ATP, that energy required in the brain. And estrogen promotes this entire bioenergetic pathway. In addition, what Ron and colleagues in the laboratory discovered is that what it's also doing is preventing the conversion of the brain to the Alzheimer's phenotype. And the Alzheimer's phenotype is the phenotype of using alternative fuels than glucose. It's switching over to using alternative fuels as if the brain were starving to death. So estrogen is creating or promoting the ability of the brain to primarily use its fuel source, its primary fuel source, glucose, and preventing its conversion to alternative fuel sources. In addition, this whole glucose metabolism and conversion into acetyl-CoA is critical for the generation of neurotransmitters like acetylcholine, as well as lipids and cholesterol. Now, why is that particularly important? Because the lipids and cholesterol maintain all your cell membranes, point one, but they're critical for maintaining your white matter. And your white matter is the component of the nervous system that allows you to rapidly transmit information across long distances and in a very rapid fashion. So this is essentially the action of estrogen in the brain, and that when there is a decline in estrogen, which regrettably I don't have time to show you, when there's a decline in estrogen in the brain, it's not as if the entire brain now cannot function. You're just down about 25 to 30% in your ability to generate energy. So how many of you lived through the past economic times? And so I'm not going to ask how many of you had a 25 to 30% drop in your retirement portfolio, but it hurt. Right? 25 to 30% drop actually hurts. In the case of your portfolio, economic portfolio, you can recover that. In the case of your bioenergetic portfolio, you have a limited window of opportunity to restore this system that allows you to convert glucose into acetyl-CoA that drives the generation of ATP, the energy-producing capability in the brain, as well as to sustain the white matter. And these have consequences, obviously, for sustaining the cognitive ability in the brain. And we also know that that has consequences for the generation of Alzheimer's pathology. And I will just quickly shoot forward to the summary slide. We're currently working on a hypothesis of the bioenergetic crisis of Alzheimer's disease, in which in the premenopausal brain, the bioenergetics of the brain are optimal, and that at the time of menopause, during the perimenopause, there's a decline in this bioenergetic system in the brain that now creates a vulnerability for developing the pathology of Alzheimer's disease and ultimately leads to both the development of the pathology of Alzheimer's disease, the symptomatology of the disease, as well as the loss of white matter and the generation of beta amyloid pathology throughout the brain. And so our goal is to obviously prevent this event, this cascade from occurring, and we have 3 therapeutic projects in the lab that now target that, and 2 specifically that target developing safe and efficacious alternatives to estrogen therapy. And with that, I'm going to fast forward and acknowledge the remarkable team that has brought you much of this research. And they're pictured here, and they're scattered around in the audience. And with that, I will thank you and take very brief questions. Yes?
lots of people stop taking estrogen. What does that, when they go through the menopause?
They do.
So what effect then does that have?
Am I on?
You got to use this one.
Okay, so it's interesting that you asked that question because we just had an article published in the New York Times, and PhoneThat is a blog, which is this is the new science, and there are, as of this morning, 375 responses from women detailing their experiences of the menopause. And it's all different. And so part of what my colleagues, Christian Pike and Todd Morgan and Tuck Finch and I are currently working on is determining which trajectory women go through during the perimenopause. Because you're right, there are about 4 different phenotypes, as we would call. Women who go through the perimenopause and they're just fine. Women who go through the perimenopause and they get better. That's a very small percent. There's another phenotype of women who have very mild symptoms and kind of tough it out. And then there's another phenotype of women who are debilitated, who suffer very, very serious consequences of a loss of ovarian hormones. And we think the reason why there is this, at least variation in those two, is that in some women that have symptoms of perimenopause, what we think is actually happening is that there are adaptations in the brain. And maybe it's what Tuck mentioned this morning. I was on the webcast. I was listening to the webcast. And we think some of those women can actually now generate estrogen in their brains. Right? So that's part of what we're actually starting to pursue. Sure.
And we'll come back for questions afterwards. Thank you very much, Robbie. Our very own Christian Pike now.
Good catch.
Good afternoon. Well, in the interest of sexual equality, we're going to move on to the men. Turns out men get Alzheimer's disease as well, and I'm going to talk to you a little bit about that today. Although I work with estrogen and progesterone, part of my lab is focused on understanding testosterone in aging men. So as Robbie Brinton was mentioning, one of the things that happens with Alzheimer's disease is the biggest risk factor is just getting old. And what we don't understand is what is it about aging? What are the normal biological changes during aging that increase our risk for developing Alzheimer's disease? Well, one of the things, as Dr. Brinton was just mentioning, was that in women it's menopause and the loss of estrogen and progesterone. Men don't go through menopause per se, but they do go through something somewhat similar. Andropause sometimes colloquially is called. And what happens is their testosterone levels decline with aging. Beginning at about age 30, you have this real insidious, gradual decline in testosterone, maybe only 1% to 2% a year, which is not much, but give yourself 20, 30 years and see what happens to your testosterone levels. And what happens is you get a constellation of different changes. You get a decrease in muscle mass, decrease in bone density, increase in fat, effects on your blood and on your heart, and importantly, on your brain. So the idea is that perhaps this age-related decrease in testosterone is associated with an age-related increase in Alzheimer's disease, and so that at least in some men, low testosterone is a risk factor for developing Alzheimer's disease. So to begin investigating this thing, one of the things we wanted to do is get away from this idea of looking at blood levels of testosterone, because that's a pretty weak indicator of what's happening in the brain. And what no one had done before is actually look what happens in the brain during normal aging and with sex steroid hormones. So a few years ago, we started this collaboration with Frank Stanzik, also here at USC, and we looked at brain levels of testosterone in men with no neuropathology at all. And so looking beginning at middle age all the way up to about age 100. And what you can see is there is, in fact, a dramatic decrease in testosterone levels in brain, normal aging brain. And in fact, once you get past the age of 80, the amount of testosterone loss in brain is much greater than what you see in blood. So the brain is particularly starved for testosterone. Now, testosterone is an interesting hormone. It not only acts directly as a hormone, it also serves as kind of a precursor or pro-hormone. And that in tissues, it is converted to other hormones. So in the brain, it's converted by this enzyme 5-alpha reductase into another potent androgen, dihydrotestosterone. This hormone also shows age-related decreases. Testosterone is also the source of estrogen in men, 17-beta-estradiol. Estradiol levels do not change in men. So what you're seeing in men is specific loss of androgens, testosterone, dihydrotestosterone. So what does this have to do with Alzheimer's disease? What we did is then compare levels of testosterone in these normal men with those that develop Alzheimer's disease. And what you can see is the men that have Alzheimer's disease do in fact have lower levels of testosterone. This is interesting, and it shows us, in fact, low testosterone is associated with Alzheimer's disease. What's not clear is whether this is perhaps a consequence of the disease, or more importantly, might it be actually contributing to the disease. So we looked at another group of men with what we called mild neuropathological changes. The brains were showing the beginnings of Alzheimer's disease. They don't have the neuropathological criteria to be diagnosed with it, but they have early changes. And so when you look at these MNC, or mild neuropathological change men, you also see low levels of testosterone. So what this suggests then is that testosterone loss is relatively early in the disease and acts as a risk factor, promoting the development of that type of pathology. So if we look at testosterone's metabolic byproducts, DHT shows the same type of relationship, although it did not meet statistical significance, whereas estrogen levels in the male brain are not associated with development of Alzheimer's. Now, I won't show you the data, but if we swap out the men and we look at the women, it's quite different, and that is estrogen levels in women. Low levels of estrogen are tied into Alzheimer's disease. So it's different for the two sexes. So where do we go from here? So what we have is a relationship in the human brain where low testosterone is tied into a risk for Alzheimer's disease. How is that working? So what we want to do is look at Alzheimer's pathology. And the two major forms of pathology in Alzheimer's are plaques and tangles. So in this dark brown is a beta-amyloid plaque. It's a normal protein that abnormally accumulates and is believed to be the primary cause of the disease. And in dark blue-black color here is another normal protein, tau. But in Alzheimer's disease, it becomes chemically modified in a way, phosphorylation, too much phosphorylation, and it's called hyperphosphorylation. And that develops into tangles. So these two types of pathology are really the hallmarks of the disease. And so in normal aging, you'll see a few tangles, not much in the way of beta-amyloid. With mild Alzheimer's disease, you see an increase in these levels. And then in select areas of the brain, particularly important for learning and memory, you see a lot of plaques and tangles and degeneration. Okay? So what we want to understand then is does testosterone regulate beta-amyloid? Does it regulate tau? Is that how it's tied into the disease? And if so, exactly how is it doing that? It's not enough just to understand that it is doing that, but it is important to understand exactly how. Because we know quite a bit about how amyloid is regulated, how tau phosphorylation occurs. But if we want to develop specific drugs that will go in and target these pathways, we have to know exactly how the main compound, testosterone, is working. So what I'm going to show you is a little bit of data looking at beta-amyloid and at tau, what testosterone does to those and how it's working. So to do this, we looked at some mice. And mice don't normally get Alzheimer's disease, but we can genetically engineer them with human mutations to develop a mouse form of Alzheimer's. So this is looking at beta-amyloid levels in the subiculum, the hippocampus, and the amygdala, brain regions important for learning and memory. And so there's beta-amyloid levels in a 7-month-old, kind of a young adult male. What we did is took another group of mice, and when they were 3 months of age, we castrated them, gonadectomized them, removed their testes. They have no more testosterone in them, or very, very low levels. When you do this, there we go, you see a robust increase in beta-amyloid. So that if you just lose testosterone, all of a sudden the brain is very permissive to the accumulation of beta-amyloid. Can we treat this? Well, if you put dihydrotestosterone or DHT or testosterone, you're able to prevent this effect. So low testosterone, up goes the amyloid, put the hormone back in, it comes back down. So how does this work? Well, I'm going to spare you all the details, but I'm going to give you a little bit. What happens is we know quite a lot about how amyloid is made, how it accumulates into oligomers and into fibrils. One of the important things that we focused on is the brain not only makes it, but it has ways of getting rid of it. It chews it up. One way to choose it up is some enzymes, and we focus on neprilysin. Neprilysin is an amyloid-degrading enzyme. It goes along and it chews up beta-amyloid. So if you've got a lot of neprilysin, you can get rid of too much amyloid. Well, we think that testosterone might regulate neprilysin. So if neprilysin levels go down, amyloid's going to go up. So when we look at the rats, and we looked at neprilysin levels, if we castrated them, got rid of their testosterone, in fact, the neprilysin levels do go down. If we put the hormone back in, they pop right back up. So to prove whether testosterone is regulating amyloid through this neprilysin mechanism, we switched over to another cell culture mechanism, and here we can see that testosterone and DHT reduce levels of beta amyloid, but this ability to reduce is lost if we put in a drug that blocks neprilysin. So what does all that tell you? It tells you that there's a specific mechanism. All androgens reduce testosterone levels, and they do so by a specific mechanism. All right? This would be important for drug design because you want to have a very specific pathway. I'm going to quickly jump to the other type of major pathology in Alzheimer's, tau. Okay? We did the same type of experiment, used some mice that were a little bit older because tau pathology follows beta amyloid pathology. And what we can see is regular mice develop no tau pathology. Here's hyperphosphorylated tau in a regular old male transgenic mouse. If we've castrated him and look at a few months later, there's an increase in kind of these pre-tangle-like cells. If we gave them testosterone, we're preventing that effect. So testosterone is regulating both beta amyloid and tau. It does this by a completely different mechanism. Again, I will spare you all the details, but I have to show you some because this is what we do. Here's tau phosphorylation. If you put testosterone on, the levels of tau go down, and there's a little graph showing it. We think testosterone regulates tau by regulating the most important kinase involved in phosphorylation of tau, GSK3 beta. Testosterone phosphorylates it and turns GSK3 beta off. It turns this off, and that allows tau to be unphosphorylated, and it does that by a pathway that involves PI3 kinase and AKT. What all of that says is that we have two different pathologies, two different mechanisms. So what we want to begin to do, now we have molecular targets that we can go after. Because we can give men testosterone. We have a problem with enlarged prostate. We have a problem with prostate cancer. Testosterone does a lot of good, but it also has potential problems. So what we want to look at is new types of drugs that will activate the beneficial effects of testosterone, but not have those effects in other tissues like prostate. So to do this, we're looking at new classes of drugs called selective androgen receptor modulators. This is a very hot topic right now in testosterone research, synthetic testosterone derivatives. And we're looking at one of them right now, MENT. We're looking at a few of them. I can't show you all the data, but I can show you proof of principle. I think we can get this done. If we look at the levator anti-muscle, a muscle in the pelvic floor, the levels are regulated by testosterone. You castrate them, muscle goes down, put testosterone in, it goes back up. If we put in our MENT drug, we can replicate the effects of testosterone. But we're not having much of an effect at all on seminal vesicle or on prostate. So we can selectively target the muscle. Will the drugs work in brain? Yeah, we think they will. We don't have all the effects on Alzheimer's pathology. That's still cooking up in the lab right now. But if we look at sex behaviors which testosterone regulates, this MENT drug not only increases muscle, it increases the sex drive in these animals that is lost when you've castrated them. Okay? So that's one way that we're going with the testosterone research. I'm going to switch to something else we've been talking about all day, and that's obesity and how everyone in this country is getting fat. What's the relationship with that with Alzheimer's disease? It turns out that with obesity, you have elevated fat, which leads to a constellation of symptoms: hyperinsulinemia, hypertension, dyslipidemia, insulin resistance, all of them tied into the development of type 2 diabetes. And these, by the way, all of them are associated with an increased risk of Alzheimer's disease. So what does fat and diabetes have to do with testosterone? Well, we know that fat leads to metabolic syndrome, and that can lead to type 2 diabetes and stroke and heart disease. Metabolic syndrome is also associated with hypogonadism. Guys that are fat have lower testosterone. If you lower men's testosterone levels, they'll get fatter, they'll develop metabolic syndrome. So we have two different related conditions: low testosterone, metabolic syndrome, both of which independently can drive Alzheimer's disease. But we think we're going to isolate a subset of men that have got both of these conditions and that are interactively working. So I'm going to share with you just a couple of quick slides on this. What we did to study this is look at what happens when we give high-fat food to our little mice. They get fatter, big surprise. We've increased their weight by about 50%. We've also induced a diabetes-like condition in them. They have elevated levels of fasting glucose, elevated levels of insulin. If we look at their Alzheimer's pathology, they also have an increase in beta amyloid levels. So high fat, a diabetes-like condition, and Alzheimer's, and guess what else? When we gave them the high fat, their testosterone levels went down by about 2/3. Their sex behaviors went down by almost a half. So they're lowering testosterone. What happens if we lower testosterone levels in the high-fat mice? Here's what happens to glucose under mice with testosterone. If we get rid of testosterone, there's a much larger effect on the diabetes, and both of these lead to a doubling, a synergistic effect on the levels of beta amyloid. Okay, so let me just wrap this up and remind you now that the human brain shows us that testosterone levels go down, that this is a risk factor for Alzheimer's disease, that testosterone regulates a couple of key features of Alzheimer's disease pathology by different mechanisms that we can target with new drugs, that these relationships are also affected by obesity, that a high-fat diet by itself increases Alzheimer's, and that high fat reduces testosterone, and that obesity and testosterone work together to promote Alzheimer's pathology. And I suggest the controversial idea that we begin to look at testosterone therapy and targeted testosterone-like drugs to treat these conditions. Thank you very much. We have time for, I think, one or two quick questions, if anyone has any. Yeah.
So how does the level of Ph.D. in females differ from the males that you study? And does estrogen do the same, or does it affect the same protein as does
It turns out that estrogen and testosterone have similar actions, but they work a little bit differently. When we look at estrogen, it doesn't affect neprilysin. It affects another similar enzyme called insulin-degrading enzyme, or IDE. Interestingly, men and women have very different levels of hormones in their blood, but in the brains, they're much similar. So men and women have somewhat closer levels of estrogen in the brain than they do in the blood. So all these hormones are acting both in the male and the female brain. Okay?
All right, well, Robbie and Christian have depressed us all so much that now Margaret Gatz is going to explain depression to us.
I don't have a pocket. Okay. Can you hold this okay?
Can we set it somewhere?
Sure.
Okay, thank you. Thank you, Dean Davison. Thank you, Dr. Davies. So I get this invitation to participate in a session on diseases of aging, and there's 4 of us. So I'm thinking, 4 diseases, and there's so many to choose from. It's even hard even to talk about only 4. And then I learn that everyone else is going to talk about Alzheimer's disease. I'm thinking, well, what should I do? So I ran down the alphabet a little further, and I got to arthritis. And I'm thinking, okay, arthritis. Among people in the audience, 65 and older, 60% of you will have arthritis. This could be a good topic. And then it occurred to me I knew nothing about arthritis. So I kept rolling further down the alphabet, and I got to depression. So the diagnostic criteria for major depressive disorder, in fact, provides a really good introduction to the symptoms that might signal depression. So in order to be diagnosed as having major depressive disorder, the person must report or show depressed mood most of the day, nearly every day for at least 2 weeks, or must report or show symptom number 2, which is a diminished interest or pleasure in all or almost all activities. And then some additional symptoms from this list so that one hits a total of at least 5 of the 9 listed. So the others also include some somatic symptoms, changes in sleeping or eating, just not being able to get going, and the self-deprecating kinds of cognitive symptoms. And a final criterion is that these symptoms can't be better explained by something medical, like having the flu or a side effect of a medication. The most recent epidemiologic figures for 12-month prevalence of different disorders show that major depressive disorder, which is the biggest component of mood, has a 12-month prevalence that goes down with age. So the 75-year-old and older are the maroon bar, the red bar being 65 to 74, and these are data from the National Comorbidity Survey replication. Now, in addition to noticing that rates of mood disorder are lower as in people who are older, please also notice another interesting thing, which is there's a ton more anxiety disorder than there is depressive disorder or mood disorders. So what else do we know about the epidemiology of depression in older adults? So in addition to knowing that it's less prevalent in older adults than in adults of other ages, we also know that depression may be under-recognized. And among the symptoms of depression, older adults are less likely to pick that mood symptom and to be spotted for depression based on the mood symptom. What's much more likely is that older adults are endorsing symptoms of the loss of interest or can't get going variety. So it makes depression in some ways much harder to detect. There's also a tendency both in older adults themselves and in their healthcare providers to regard depression as something that's sort of normal with aging and therefore not to go after it and treat it aggressively. One more note, which is that older adults, more than adults at other ages, tend to endorse quite a lot of symptoms of depression, but not enough to meet disorder, to be diagnosed as having major depressive disorder. And if one looks at the risk and protective factors for symptoms of depression falling short of being diagnosed as major depressive disorder, they behave pretty much the same as major depressive disorder and respond similarly to treatment and in fact warrant detecting and treating just the same as major depressive disorder. So the other idea people have is that there's a lot of depression in older adults. So it comes something as a surprise that the rate is as low as it is. And this led Lissy Jarvik to write about an amazing paradox. What's amazing is she wrote about it 35 years ago, where she says, so why, in the faces of losses that occur with age, rising frequency of somatic illness, physical debility, sensory deficits, reduction in mental agility, economic deprivations, curtailment of social roles, death of friends and family, so why, in the face of all of that, isn't every older adult depressed? So to answer this, I believe, requires a lifespan model. And in this lifespan model, I want to point out a couple of things. One is to look developmentally at the occurrence of depression. And in all the studies I can find, on average, a little more than half of older adults who meet criteria for major depressive disorder, it's the first time in their lives they've experienced depression. And then somewhat less than half will have had an episode earlier in life. Now, this doesn't help at all answering Lissy Jarvik's paradox. It takes us along a little bit further to also saying, let's start looking at the risk factors and the protective factors for depression, and let's also situate these developmentally, and then maybe we can begin to understand the patterns that we're seeing. So Amy Fisk, Julie Weatherall, and I put together a review of depression. This is freely available online. And this is a model that we suggest for thinking developmentally about risk and protective factors. So we show the risk factors above the line in the pale blue, and we show the protective factors below the line in the cheery yellow. And these are sequenced close to developmentally, showing when in someone's lifespan this particular risk factor for depression might occur. Yet all of these are risk factors for depression in older adults. Some of them can also be risk factors for depression at younger ages. And here we do begin to see a couple of things. And one is what starts to be an answer to Lissy Jarvik's question. and that's found in these protective factors, is that with age, there are increases in things such as being able to manage stress better, being able to maintain emotional equilibrium, and that these protective factors may start accounting for why, given that long list of stuff that happens more and more with age, older people are depressed less, not more. I refer to this in the title of the talk as a lifespan developmental diathesis-stress model. And the diatheses are those things like genetic or personality factors. These are propensities that one brings to the situation. The stresses are the environmental or the external occurrences like bereavement. And in this particular diagram, both diatheses and stressors are included. So research shows that different ones of these risk and protective factors are more important when the onset of depression is in early life or in later life. And one thing in particular that comes up is that older adults with late onset depression are far more likely than older adults who had had depression earlier in life to have cognitive deficits, particularly frontal-related cognitive deficits, executive function. They're far more likely to have neurologic findings, including white matter hyperintensities on neuroimaging. And when this was initially discovered, Alexopoulos called this vascular depression, although he now prefers the more accurate but clearly more cumbersome title of depression executive dysfunction syndrome of late life. Now there's a disease to avoid. Huh? So we've been doing research in Swedish twins, and some of our research supports this general picture, and I want to walk you through this model. So we looked at twins where the index twin has major depressive disorder, and the blue line says, what's the odd of the other twin also being depressed? And if the first onset of major depressive disorder was early in life, there's quite a high risk that the twin will also develop depression. After about age 35, the risk to the twin becomes non-significant. It's not important. But then you have the converse. And the red line shows, given the age of onset of depression in the index twin, what's the likelihood that the other twin will have vascular disease? And what you see there is this upward trending line, so that if the first onset of depression in the index twin is after age 45, the co-twin has an increased risk of vascular disease. So this goes really nicely with this vascular depression hypothesis. Can't leave Alzheimer's disease out of this altogether. So here are data from Jessica Brummelhoff's second-year project. In this case, we're looking at twins where one has dementia and the other does not have dementia, and we ask, what is the likelihood that there's a history of depression? And in the red boxes, we see the twin with dementia, and we see it's twice as likely that that twin will have a history of depression, and most of that depression occurred within 10 years of the onset of the dementia. Whereas with the twin who did not have dementia, there's less likely to have a history of depression, and it most likely occurred greater than 10, greater than 20, or even 30 years before, suggesting that in some cases what's going on is there's a form of depression that may be sort of part of the same syndrome that eventually is going to manifest itself as dementia. Now I'm going to flip really quickly through a behavioral model because I don't want to leave out the importance of stress in people's lives. You wouldn't want me to leave stress out of your life. So in the blue box, a really well understood theory of depression is that there's a low rate of positive outcomes in one's life. In older adults, this often comes with limitation of activities, which can be brought on by changes in social roles, like loss of the person you used to do stuff with, long-standing vulnerabilities, approaching life with the glass half empty, changes in health, cognitive ability, and then a really negative cycle starts where then the individual starts blaming him or herself for the mess I'm in, and this maintains depression brilliantly. Now, the reason for pointing this out is that there's a very good news response to depression, which is depression responds very well to treatment. And the psychotherapies that have been shown to work well with depression pick up from that cycle that I just showed you and attack both increasing the level of activities in people's lives and trying to change those cognitions that keep people caught in a bad cycle. These approaches also are effective with caregivers who suffer depression and with older adults with dementia who suffer depression. So, in summary, with age and with accumulating experiences, there's a shift in the kind of stressful activities that older people, there's a shift in the kind of stressful events that older people encounter. And this shift in kind of stressful events is more likely to lead to curtailment of activity. And in addition, there appear to be some brain changes that clearly account for some portion of late-life depression. At the same time, there's an increase in stress management and emotion regulation skills, which counterbalances these risks. And for those who do become depressed, there are effective treatments, and it's important to bring those treatments to bear and bring those treatments to bear early before someone gets stuck in that rather bad rut that I showed you. Thank you. Okay.
So you may have solved the dilemma I've had for years about major depressive disorder, less prevalent, at least in cross-sectional studies, in older people than in younger. But for depressive symptoms, more prevalent? Those are more prevalent among older adults?
Now, if you take the entire lifespan, the major depressive disorder has this distribution, and depressive symptoms have this distribution.
Okay. So sort of highest in the middle part. Okay. On the major depressive disorder. Okay. Does that mean that in the DSM framework that among older adults you have lower incidence of the first two, the ones either or have to be present, low mood, and that second one, and more of the more optional ones from which you only need to have 3 or 4. You see my reasoning?
Yeah. Number 2 in that list, the loss of interest, is common in older adults, but it's harder to spot. And so one of the things that we're doing right now is taking which symptoms people check on a self-report symptom checklist and seeing which people physicians correctly catch as depressed. And the hypothesis we're running on is that we actually know if we took a quick pilot peek at the data that this is what we hope is going to keep happening, is physicians will spot it better if mood is one of the symptoms.
I'll talk to you later.
Time for one more quickie.
Oh,
Margaret,
okay.
Another quick question? Okay, yeah.
Margie, could you explain again a little bit the relationship between late onset depression and dementia? And are you saying that late onset depression can in some way be a precursor to dementia?
My thinking is that the late onset depression is not a precursor or a risk factor so much as it is an early sign, a prodromal symptom. But there's a huge argument about this. And particularly the group at Rush in Chicago strongly believes that depression is a risk factor. And I think, based on our data, what it looks like to us is, at least in some people, it's part of the same syndrome. And we see this in twins with what the sequence of symptom is. So if both twins develop dementia, frequently if depression comes first in one, it comes first in the second. There's some very familiar kind of pathway of clearly something's wrong, and then it begins to look like depression, and then it pops up to be dementia. And we think that's what we're capturing in our data with the people whose depression was closest to the dementia onset.
Thank you, Maggie. Okay, hello again. Like Maggie, I also noticed that we had only one disease. So now you've added depression, thank you. And I'm going to try to add Down syndrome on here and Huntington's disease. So we will, in fact, get our 4 diseases under our belt before we're done. So I never actually meant to study neurodegenerative diseases. I had no interest in them. I was, if anything, interested in cardiovascular and muscle diseases. But I got dragged kicking and screaming into this all the way, because we discovered a gene that we originally called ADAPT78, and this was a gene we discovered during adaptation to oxidative stress, the kind of stuff for those of you who were here this morning. And we found this gene in 1994 when I was working in Albany, New York. It's on chromosome 21 in human beings. Here's the genomic DNA for those who are interested. It has 7 exons. You can mix and match the parts of this gene and make different products from it. Here are the 3 major protein products that are formed from it. And it's a very interesting gene that does all sorts of things in adaptation that we're interested in. Here's a lesson, a tale for the students in the audience. We originally called it ADAPT78, as I said. I'll use this one. ADAPT78. And because it was in an adaptation. Another group found it in the same year by genetic linkage analysis, and they called it DSCR1, which stood for Down syndrome critical region 1, the gene linked to Down syndrome. We eventually, after years and years, and 21 other groups getting involved in the research, and at least 21 other names, eventually convinced everybody to call it ARCAM, which is regulator of calcineurin 1. And this whole paper, the whole front page, is just the names and the places where people came from. This is political science. It's an interesting exercise. But anyway, ARCAM 1, it's important because the gene was being lost. We actually saw 2 studies within a year of each other, repeats of each other in different journals. They didn't know that they did the same work. It was 2 different names. So the ARCAN gene is actually a family of genes. There's ARCAN1, ARCAN2, ARCAN3. Lots of interesting things go on. I'm just going to talk about ARCAN1. Here's where we found it in stress. Here's after 90 minutes of stress, 5 hours of stress, and what the darker the blob here, the more the gene is being expressed. So there's a lot of ARCAN being expressed in stress adaptation. So as I say, we now call it ARCAN1, regulator of calcineurin. So what on earth is calcineurin? If this is a regulator of calcineurin, what does calcineurin do? Calcineurin is what's called a phosphatase. So if you have a protein that has a phosphate group, shown by P over here, on it, if you have a phosphatase, you can remove that phosphate. And that's what a phosphatase does. It's opposed by a kinase, and a kinase puts the P back on. Calcineurin's what's called a calcium calmodulin activated protein phosphatase. And it's involved in all sorts of things in cells. In stress, what it actually does, what we've been able to show, is that in stress responses, there's an off switch for the stress responses, and that actually is calcineurin. By our protein being overexpressed in oxidative stress, it inhibits calcineurin, prevents calcineurin turning off the adaptational response, so you get a longer and stronger adaptation than you would otherwise get. It's a little counterintuitive, but that's how it seems to work in oxidative stress. In other cases, however, how might ARCAN, regulator of calcineurin, when I say regulator, it's almost all inhibitor of calcineurin, how might it work? Well, it turns out the calcineurin has major roles
in all of these transcriptional pathways. And what I want you to take home from this is simply that this is the cytoplasm of a cell, this is the nucleus of a cell, and changing activities inside a cell or to which a cell has to respond are mediated by these transcriptional regulation pathways, lots of key ones of which including things like NF-kappa B and MEF2 and the NF-AT pathway and the ALK pathway for those involved in those are regulated by calcineurin. So in turn, regulator of calcineurin, our protein, regulates all of these pathways. If we look at that, oops, I have to go back the other way, sorry. If we look at that, then what does it do physiologically? Well, deactivation of stress responses, as I said, for calcineurin. So our protein does the opposite. This NF-kappa B story, the NF-AT. And here we'd have two interesting characters, tau and Huntington. Now, you heard a lot from Christian Pike about tau. I'm going to tell you some more, and I'll tell you a little more about Huntington too. Also involved in T-cell activation, muscle hypertrophy, apoptosis, that's a way that cells can die, activation of nitric oxide synthase, and actually also in neuronal development that we won't have time to talk about today. And I showed you those. So it's expressed in neurons. These are human neurons, and where the arrows coincide is where it's expressed. These are rat neurons. So in both rats and humans, it's expressed in the neurons, not in astrocytes or glia to any great extent. I want to talk first about Alzheimer's disease. We saw ARCAN1 being expressed very rapidly for very short periods of time, for hours, in stress responses, and then being turned off. That seems fine. But now that I've shown you all these other pathways that calcineurin regulates, and that therefore ARCAN can regulate also, it would not be a good idea to turn off many of those pathways chronically or permanently to any great extent. So what if ARCAN were turned on chronically, we asked? And what we thought of right away was, as I showed you, one of the things that calcineurin does is to dephosphorylate the tau protein. And as Christian showed you, tau gets hyperphosphorylated in Alzheimer's disease, and that leads to neurofibrillary tangles and all sorts of other Alzheimer's disease problems. So we wondered if, in fact, in Alzheimer's, things like reactive oxygen species or free radicals as part of the inflammatory response, A-beta, tau, phosphorylated tau, whether ARCAN could be involved in that. So we hypothesized that chronic expression, overexpression, could be associated with tauopathies, diseases in which tau is hyperphosphorylated, such as Alzheimer's disease, and by the way, Down syndrome. And I'm cheating a little bit because I won't actually show you Down's data, we don't have time, but it's the same as Alzheimer's. So here, when I came here back to USC in '96, we started looking at Alzheimer's patients, and these are post-mortem samples from Alzheimer's brains. And you can see here, this is the Alzheimer's, and the gray bars, the black bars are controls. And ARCAN1 is overexpressed about double in regions of the brain that show Alzheimer's disease and not affected in the cerebellum that's not affected by the disease. There's an even better correlation with neurofibrillary tangles, which you might expect if we're right about the progression of what's going on. Now, interestingly, you can also turn ARCAN1 on not just by oxidative stress or by inflammation, but also with A-beta. And I apologize, this slide goes the wrong way. If you add A-beta, here's no A-beta, a little A-beta, adding more A-beta to cells, you get more and more ARCAN1 expression. So ARCAN1 responds to A-beta. These are two of the things that are going on in Alzheimer's disease. Reactive oxygen species from inflammatory processes, A-beta produced from amyloid precursor protein, causing ARCAN1 overexpression we see, which can lead to hyperphosphorylated tau because calcineurin is inhibited, and so calcineurin can't dephosphorylate tau anymore. So this should lead to tau accumulation. It's worsened by the fact, actually, that the proteasome, this proteolytic enzyme that I talked to you about this morning, degrades tau very well when it's not phosphorylated. When tau is phosphorylated, it becomes a lousy substrate. So it actually accumulates all the more. So now we're seeing several things that might lead to tau accumulation. And that's shown here, where calcineurin inhibition chronically can lead to tau hyperphosphorylation, decreased proteolysis, and accumulation, and therefore neuritic plaques and neurofibrillary tangles. Another part of the story, Christian also mentioned GSK3-beta. We've really got to talk more, Christian. GSK3-beta, which phosphorylates tau. It's also called tau protein kinase. And we see that ARCAN1 actually induces GSK3-beta and also induces phosphorylation of tau. So, and this is in a cell culture model in which we've transfected cells with the ARCAN1 gene, and we can overexpress it whenever we want. And when we overexpress it, these are the results. So from that, one could then come up with a another level of explanation perhaps, in which tau is being hit from both sides. You can't dephosphorylate it because you're inhibiting calcineurin, and you're overphosphorylating it because you're making more GSK3 beta, the enzyme that phosphorylates it, and so on down the road. Now, Alzheimer's disease is clearly a multifaceted disease. We don't have the answer here as to what causes Alzheimer's, but I think we've got some pieces of the puzzle. You put that together with Christian's data and with data from Tucker for many, many years of studying A beta and its effect, and I think you start to see a lot of relationships here. And of course, with Robbie's work on hormones, there's a lot of interesting things for us to start looking at. So we wanted to see what happens if we overexpress RCAM1 in a transgenic animal. So these are all transgenic animals that we've got, and here's one that we're looking at overexpressing. This is a Gallia stain looking at an Alzheimer's brain, and you see all these dark, patchy bits that are really bad. In a wild mouse, you don't see anything, and here's our transgenic mouse. It's not quite the same as a human Alzheimer's brain, but there's clear similarities, and this is work that we're following up on as I speak. All right, quickly switching to Huntington's disease and calcineurin, and therefore RCAN. Huntington's disease is a disease in which there is a repeat, a CAG repeat, cytosine, adenine, guanine repeat in the DNA of these patients. That encodes the amino acid glutamine. We normally have in normal Huntington protein about somewhere around 20 polyglutamines in a row, a long chain of glutamine, glutamine, that's okay. But when you get over about 28, you start having disease symptoms, and that's a real problem. It's age-related. If you have 120 repeats down here, you'll have the disease onset in infancy. If you have only 28 repeats or so, you might wait till you're 70 before you start seeing disease symptoms. So there's a clear age dependency and a dependency on the number of repeats. But there's a region in here, which is shown in this table in blue, where if you have an intermediate number of repeats, 28 to 35, or even 36 to 40, there's not really any prediction of how severe the disease will be or whether you'll have disease symptoms early or late. So there's something else going on beyond the pure number of polyglutamine repeats. And what we wondered, we started putting some other things together from other researchers' data. Could it be that RCAM11 is, and there's a specific form 11 long, is diminished in Huntington's disease, the opposite problem of Alzheimer's disease? And we started thinking that because in model systems, Huntington appears to be only minimally phosphorylated, and increased phosphorylation using two different methods of doing it is protective against the disease in model systems. So we started wondering, well, you could do that if you weren't inhibiting calcineurin normally. So we looked in Huntington's disease patients, and I apologize, it's hard to get the right number of samples to do analyses. So here are 5 non-Huntington's disease patients. You see lots of RCAN. Here are 5 Huntington's patients, 5 other non-Huntington's patients, 5 Huntington's patients, and here all of them aggregated. And you see about a quarter to a third of the level of RCAN expression in Huntington's disease that you see in normal individuals. Switch now to a cell culture model. This is a cell, a neuronal cell that we have in culture that mirrors, or mimics, I should say, some of the disease effects in Huntington's disease. It expresses, we have one that expresses 15 polyglutamines and another one that expresses 120 polyglutamines. This one's expressing the 120 polyglutamines, and when we switch it to a higher temperature, to growth at 39 degrees, it starts producing this mutant protein, and it starts killing cells. This is cells killed. So we start out with no cells killed, then we go into a couple of thousand cells killed. If we, in the same cell, transfect that cell with the RCAN gene, so we put the RCAN gene into that cell as well, and we turn on RCAN, then what we find is that we get protection. So this is number of cells killed, this is percent protection. We can protect 90% of that cell killing by turning on RCAN. So that's an impressive, for us at least, protection in this case. There's a little problem with that model, and there's a temperature shift you have to deal with. So this is another way of doing the same experiment in which we can actually turn on differentiation in these cells by a hormonal mixture. And so in these cells, when we turn on differentiation by a hormone mix, this is the low cell count you get with the cells that are not expressing RCAN. When we express RCAN, lots more cells grow by cell division. So this is very effective. Still have 5 minutes left? I can slow way down. I can tell jokes. Wow, my life story is coming before you. I didn't realize I had that. All right. In contrast to tau protein, as well, as another side to this, lots of little bits to this story which would make it fascinating, I think. Huntingtin actually has to be phosphorylated in order to be degraded. So just when you think you've understood a principle, which is that proteins like not to be phosphorylated so they can be degraded, Huntingtin comes along, and Huntingtin says, no, I want to be phosphorylated if I'm going to be degraded by a protease. So it's the exact opposite again of tau. And since we've got low levels of ARCAN in Huntington's disease, which means that you're not opposing calcineurin, which means that calcineurin is dephosphorylating all the huntingtin it can find, in that instance, then, it makes huntingtin a poor substrate for degradation. Since you've got a mutant huntingtin, which has all these polyglutamines, it likes to aggregate. And the aggregation of that huntingtin is clearly involved in the progression of the disease and in the disease severity. So that not being able to degrade huntingtin because you're not inhibiting calcineurin appropriately, as normal people do, is again a reason that you can accumulate huntingtin, the mutant huntingtin protein, and worsen the disease symptoms. So what we're wondering is, beyond that, is it now, and that's where I've sort of been aiming at for Huntington's disease, is it possible that ARCAN fills the gap in explaining why those people with an intermediate number of repeats of polyglutamine, why some of them have high disease penetrance and some very low disease penetrance, why some have early symptomology, and why some only show symptoms late in life. It's possible that it might be related to the level of ARCAN expression in each of these different kinds of patients. So now what we want to do is to take a look at a survey, which is very hard to do. It's hard to find the number of patients with Huntington's disease who are willing to be studied, and it's not an easy thing. But we're trying to get to that to see if ARCAN actually may explain the disease penetrance and the disease severity. So ARCAN. One form of it, ARCAN1/4, may potentiate stress adaptation. I didn't mention to you, but it's actually a specific protein, form of the protein, ARCAN1/4, rather than 1/1 or 1/1 long or all these other modification names. ARCAN1/1 may promote tauopathies like Alzheimer's disease and Down syndrome by preventing tau dephosphorylation and increasing its phosphorylation. And what's that? 3. I'll have to slow down even more. And thus it may promote Alzheimer's disease and Down syndrome, and perhaps other tauopathies. We're now interested in finding other. There are some weird tauopathies out there with very strange-sounding names that I can't remember, that are very, very rare, but share this problem. And if it turns out that they're linked through ARCAN, it would be even more exciting for us. A lack of ARCAN might allow aggregation of huntingtin protein, and that may help to explain part of Huntington's disease. What I haven't shown you today was some new work that we're doing looking at mitochondrial functions, where it turns out that ARCAN also directly and strongly affects mitochondrial functions. In particular, it affects a particular protein in the mitochondrial membrane that's involved in ADP, ATP translocation. And that links with what Robbie was talking about a little earlier, so we may have some interesting things to talk about. But no time for that, but that's looking very promising. We're just putting a paper together on that. And finally, what I haven't shown you at all is it can actually inhibit stem cell differentiation. We're doing quite a bit of work with stem cells, and it turns out that ARCAN strongly inhibits their differentiation. And there's another ARCAN form 1/2. There are actually several more, but one that is seen only in fetal tissues, never beyond the fetal state. We have no idea why that is or what it means, but it must mean something. And we think that's a good place to look. When I found the gene in 94, that was in Albany, New York, when I came back here in 96, pretty much all the work that we've done here on ARCAN has been pioneered and has been led by Professor Gennady Ermak. Gennady, would you stand up and please be recognized? And really, it's Gennady's work that I'm showing you today, not mine. And he's shy, but there he is. And Catherine Harris, who was a former graduate student, who's not here today. And I thank you for your attention. So I'll allow myself one or two very quick questions, and if my other panelists would come back up, we could sit up the front and answer any other questions there might be. Yes, please.
For all these proteins like ARCAN and tau proteins, are they expressed in all brain neurons? And why does it only show like Alzheimer's disease?
So the first part of the question was, is ARCAN expressed in all brain regions? Or Gennady, what would you say to that? Yes, Gennady says. That's the definitive answer. We find vanishingly low levels in astrocytes and glia. Not to say that we don't find any, but we're really very, very low.
And so why does it only kill the neurons that's related to...
Great question. It's a great question. I don't have an answer. Yeah. Okay, let's open this up for the whole panel then. So questions, please, from the audience. We can go all the way back to the beginning. You can go outside of Alzheimer's disease now. We've opened it up to more diseases. Yeah.
I actually have two questions. One of them has to do with beta amyloid, and I wondered if in the course of your research that you have also looked at primary as well as genetic amyloidosis, and if there's been any interface and treatment that you've recognized or commonalities that, other than the amyloid, obviously.
Let's start with the first one first. I haven't, but Robbie or Christian, do you want to take that?
There's just a very few percentage of cases that involve familial mutations that lead to an excess amount of beta amyloid. And as far as we know, what the mutations are doing is just increasing either the amount of total amyloid or its ability to form those plaques. And most people don't think that there's a specific difference. It's just those mutations make it more likely to occur. So we haven't specifically looked at the two, but most people believe that the amyloid accumulation is similar, and the results of it are the same.
Thank you. And the other question has to do with the interface or outcomes related to major depression and bipolar disorder. Again, are there any intersects in that or commonalities in treatment?
With bipolar disorder and major depressive disorder, bipolar disorder tends to be treated with a combination of medication and psychotherapy, and the need to include medication is fairly strong. Major depressive disorder can be thought of as actually quite different in the sense that it's less driven by genetics, and one can treat with psychotherapy or with psychopharmacology or with a combination.
Other questions? Yeah. Thank you.
Along those lines, the depression study that you were talking about earlier, what indications, if any, did you find with adults who were taking antidepressants?
When we look at the relationship between dementia and depression, we include with people who are depressed people who are already taking medications and who may not at that moment be depressed, but in that sense are part of the group. It doesn't exactly answer your question, but in what I showed you, the people are folded in together. Some interesting comparisons of different treatments for depression include the observation that with older adults, the follow-up and maintenance of a depression-free life tends to be a little higher in those who receive psychotherapy and not drugs alone. Sort of a teaching life skills idea.
All right. One question up the back.
Yeah, I had a question for Dr. Brinton about the estrogen as a regulator of glycolysis consumption in the brain. Would menopausal women be particularly susceptible to like a negative effect in any sort of carbohydrate restrictive diet? As in, for example, like I know with the Atkins diet, they advocate going into a state of ketosis. So would that be particularly negative for a menopausal woman?
Well, you ask a very good question. If I'd known you were going to ask a question, I'd have asked for more time.
And we would have given it to you.
So actually, Jia Yao in the laboratory, who has just almost received his Ph.D. degree, did a very interesting experiment, which we've not published yet, and that was very surprising to us. And that is he glucose-deprived mice. He glucose-deprived normal mice and Alzheimer's mice. Now, the caveat to all of this is that they were young, right? And so what you're really asking about is an older cohort. But in the younger mice, it turned out that reducing glucose in the, we'll just talk about the Alzheimer's mice, had a positive benefit. But what he'd also shown earlier is that these mice already had a brain that had switched over to kind of the starving brain. So in that case, glucose-depriving animals, triple trans or the Alzheimer's mice was beneficial. The question is if you take a brain that has already switched over to kind of a ketone brain, are you actually going to have benefit? And we don't really have the answer to that question yet. But essentially, there should be some modest benefit because that's what that Alzheimer's brain is using as fuel. The problem is that fuel is never an efficient fuel, right? So you're always using, the lowest grade fuel, and that's going to have consequences in the end. So, from my kind of knowledge of the system, I would predict that would not have benefit. The kind of Atkins diet would not have benefit for very long. And in fact, that's true. There's a clinical trial that has demonstrated that it actually had no statistical benefit, and whatever benefit was there actually was very short-lived. I would like to bring up an issue.
And in the interest of giving Robbie Brinton more time, we'd love to hear.
I do think it's interesting that there seems to be this very close parallel between loss of reproductive hormones, because Christian showed you this increase in beta amyloid generation if the mice had lost the ability to synthesize testosterone. Well, his group and our group and a number of other groups showed earlier that if you remove the ovaries, the same thing happens. So there seems to be this remarkable convergence around the loss of ovarian hormones, and testicular hormones, that essentially lead to the risk, if you will, of developing Alzheimer's pathology. And I think the convergence, again, of these hormones on the bioenergetic system of the brain and on regulation, for example, of glucose metabolism, all give us, I think, remarkable insights into potential mechanisms for late onset. And so one of the things that I suggested to Christian is that he and I have got to write a review after we write this program project renewal. So it's I think it's really brings up the importance of multiple investigators approaching a question from multiple perspectives and the power that this convergence upon the bioenergetics of the brain has. And that also brings up the whole point about bioenergetics and risk of depressive.
Mm-hmm. Yeah. Right. This is a hypothesis that estrogen's in this same cycle.
Mm-hmm.
Data analysis occurring as we speak.
Very good. Thank you all very much. I'd like to thank Sharon Lynn, who I didn't thank this morning, who was involved in some of those first mitochondrial studies when she was a graduate student with me and is now working in Tuck Finch's lab years later. I'd like to thank my three panelists,
thank you,
co-panelists, and thank the audience.